What we're seeing here is the graded and the action potential. We have learned at length. Um, Local potential are graded, which is known as the graded potential, meaning that they vary in magnitude according to the strength of the stimulus. Graded potential are decremental. That means they decrease with distance. That is, they get weaker as they spread from one point to the stimulus. They decline in strength. Occurs partly because sodium spreads over under the plasma membrane. Depolarization happens. Sodium, spreads across the plasma membrane, and potassium flows out and reverses the effect of sodium inflow. And these together causes the decrease in the strength of the in case of graded potential. And graded potential is reversible, meaning that if stimulus decreases or ceases, the plasma membrane will go back to being at the resting stage. And of course, local potential can also be excitatory and inhibitory, as we have learned before. The other part, which is the action potential, we learned that, right, as well. One very important thing to remember about action potential, which is it goes through the three phases, we know that, which is depolarization, repolarization, and hyperpolarization. But it follows all or none rule. That means if a stimulus depolarizes a neuron to threshold, that is, if it goes to threshold, the neuron fires at its maximum voltage, that is positive 35 millivolt. If threshold is not reached, if it is not reached, the neuron does not fire at all. And this is once the threshold has reached, it is at that point, there is no return. It will go through the entire process of action potential, which is depolarization, repolarization, and hyperpolarization. And this is once the one neuron is affected, the rest of the neuron in the nearby is also affected at the same time. All or none principle means once it's initiated, once it reaches the threshold, it will follow the entire process. Action potential is non-decremental, meaning that it does not decrease with distance. That is, it does not get weaker with distance. The last action potential at the end of the nerve fiber is just as strong as the first one. So when an action potential starts in one neuron, and basically when it ends near the axonal terminal, the first neuron that fires, the strength of the first neuron and the last neuron is equal. Again, this is true for both graded and action potential, that it is, it causes electrical excitability of the plasma membrane. One other difference between local and action potential is action potential is irreversible. That is, if a neuron reaches a threshold, action potential goes to completion. It cannot be stopped once it begins. It is very similar to that of, if you think of a gun, firing of a gun, as the trigger is squeezed, you squeeze the trigger, the gun either fires with maximum force or it does not, right? So if you touch the trigger lightly, slightly, it will either most probably the fire will the bullet will not come out, but with a particular amount of pressure, which is called the threshold, in this case, the bullet will come out. So, but once the threshold of your finger is given to that gun, there is no return. The bullet will fire, and it will go out of the barrel. Okay, so once the trigger is pulled to its threshold, at that point, the bullet always leaves the muscle at the same velocity, okay, the same velocity. So basically, these are a couple of differences between graded potential and action potential, and it is important to remember the differences between the two. Remember, both graded potential and action potential are both required to make the plasma membrane excitable. This takes us to the next part, which is the mechanism of action potential. We have learned most of it, and we are going to continue doing that. If you look into this diagram, this is the plasma membrane, and this is outside of the cell, and this is inside of the cell. There are two gates in here. One is called a sodium activation gate. The other is known as sodium inactivation gate, and those are for sodium. And potassium only has one gate, which is this gate in here, which is known as the potassium gate, okay? At the resting state, remember, all gate, whether it's sodium or potassium channels, are closed, okay? So if you look in here, the activation gate is closed, and the inactivation gate really doesn't have much function at the resting stage, but the activation gate is of the sodium is mainly responsible for the entry of sodium. Very important to understand this. Sodium can only enter when the activation gate is open, okay? But at the resting stage, when the cell is not firing or not excitable, at that point, sodium is not entering through the activation gate because the activation gate is closed. Potassium is also closed at the resting stage. So as you can see, all gate sodium and potassium channels are closed at the resting stage. When the sodium permeability increases, which means like whether it's whether there is a stimulus that comes in and which causes the opening of the activation gate. So you can see in here, sodium then rushes in from outside to inside of the cell. So here we are talking about only two ions, remember, sodium and potassium, as we have so far. And in here, you can understand the inactivation gate at this point remains closed. That means potassium is unable to go outside. This is a phase of depolarization. So when depolarization has started, sodium is moving in because the activation gate of sodium is open. The activate or the gate of potassium is closed, so potassium cannot come out. At threshold, which is a critical level of depolarization, which is negative 77 to minus 50, at this point, when sodium is coming in, we are calling that part depolarization, right? So depolarization of phase when sodium channels are open. So once it hits the positive 30, sodium has moved in. It's important to understand that the at positive 30 millivolt, which is the part when depolarization stops and it goes into repolarization. Why it happens is at this part, the inactivation gates of sodium is now closing. Sodium channels closing and potassium channels are opening. So at this case, the sodium inactivation gates are definitely at this point sodium inactivation gets closed. Membrane permeability to sodium declines. It's going down. Trying to reach its resting potential. Sodium is coming in, but remember it is the activation gate in the process is trying to close so that sodium can no longer come in from outside to inside. The second phase, the other part of the story is at this point, the potassium gate is open and potassium is coming out. So this and the balance of these two actually leads to repolarization of the entire of both these ions, a combination effect of it, that in the level of positive ions inside is basically decreasing, and it's trying to come back to its resting phase. Hyperpolarization, sodium gates remain open. At this point, the sodium gate is open, causing excessive efflux of potassium. So potassium actually is coming out in large numbers. And remember, potassium is also positively charged. So this is leaving at this point, and sodium, which is positive, is not coming in. The combination effect is that the neuron at this point goes into hyperpolarization, which means it goes goes below the -70, which is around minus 85 or minus 90 millivolt, which is hyperpolarization. So at this time, both the gates are of sodium are closed, but you know, potassium gates are closing. But in the process of the closing, on the process, some of the potassium ions are also leaking out. As a result, before it goes into the resting phase, some more potassium ions move out, which causes the hyperpolarization effect to take place. So this slide actually summarizes all the four stages, which is the resting phase, depolarization, repolarization, and hyperpolarization. So just to show you, this is a slide to give you an overview of the entire action potential process. So let's do this again. This is the plasma membrane. As you can see, this is the extracellular fluid. This is the intracellular fluid. And as you can see in here, this is the sodium channel. This is a potassium channel. Sodium channel has two gates, as we said. This is the activation gate. This is the inactivation gate. And potassium just has one gate, which is known as the potassium gate. So this is the resting stage at which the activation gate is closed. Voltage-gated sodium channels are in the resting state, and voltage-gated potassium channels are closed. So once depolarization starts, that is depolarization to threshold, which is negative 55 millivolts, this causes opening of the sodium channels, activation gates. The activation gates are now open. Sodium is moving in. The inflow of sodium further depolarizes the membrane until its polarity is reversed. So it moves in, goes into threshold, and then goes to a point of positive 30. So if you see this graph, if you remember what we learned in action potential, this is the resting potential, and the sodium ions are moving in, crosses the threshold, crosses 0, and goes up to 30 millivolt. Okay, this is the depolarization phase. When it hits positive 30, at that point, repolarization phase starts. What is happening? Slowly and steadily, depolarization also opens voltage-gated sodium potassium channels. At this page, the potassium channels are opening. Activation gates of sodium are closing. Okay, in the process, as you can see, repolarization has started. Or at the same time, potassium channels in activation gates are closing. So this, the activation gates are closing, inactivation gates are closing. At this point, sodium can no longer come in, but potassium actually is going out from inside to outside. So at this phase, you can see the repolarization has taken place, shown in red. And then what happens in the process, what happens after repolarization, the next step is hyperpolarization, which in the outflow of sodium restores the resting membrane potential. Sodium channels inactivation gates are opening, and potassium channels are closing. So potassium channels are closing. By the time the potassium channels are closing, more and more potassium channels are ions are coming out. which causes hyperpolarization of the membrane. So we have done this multiple times, which is threshold, rising phase, overshoot, falling. These are the different names of depolarization, repolarization, and hyperpolarization. Now we are going to go over two other, terms that is part of the action potential. One is known as the absolute refractory period and the relative refractory period. Okay, so let's understand. If you look in here, you will see that this phase, when it goes from the resting potential, depolarization, repolarization, this entire process of action potential, time from opening of the sodium activation plates until the closing of the inactivation gates that we just learned, is known as the absolute refrain. Entire process of action potential has to happen. Until this entire process is complete, the next stimulus, even if it comes in, does not affect the membrane. That is known as the absolute refractory period. This is actually followed by a relative refractory period in which it is possible to trigger a new action potential, but only with an unusual strong stimulus. This is known as the relative refractory period. Usually a new stimulus cannot with a normal strength, but if that is, if it is a tremendously high amount of stimulus, then maybe a new stimulus can start. This is, the relative refractory period is the interval followed by the absolute period. As you can see, this is absolute refractory period, which is followed by the relative refractory period. Sodium gates are closed, potassium gates are open, repolarization is occurring, and if the next stimulus is even stronger and it's of huge strength, only then the relative refractory period will take place. The absolute refractory period lasts from the start of the action potential until the membrane returns to the resting potential. That is for as long as the sodium channels are open and subsequently inactivated, that we just learned. The relative refractory lasts until hyperpolarization ends. So you can see until here, which is the hyperpolarization ends, till then the relative refractory period can take place. Okay, so next we go into which is signal conduction in nerve fibers. So if a neuron is to communicate with another cell, we know that a signal has to travel from one end to the other, right? So this is important to understand that these are happening in milliseconds and the movement of ions is taking place as it goes through. So this conduction is different between myelinated neurons and unmyelinated neurons. So we'll start with first unmyelinated neurons, which is relatively simple and easy to understand. An unmyelinated fiber has voltage-gated channels along the entire axon. So there are many, many voltage-gated channels along the entire axonal length. When an action potential occurs at the trigger zone, which we know is the axon hillock, so when at the trigger zone, when action potential takes place, and unmyelinated has voltage-gated channels along the entire length. We remember when a stimulus is taking place, axon hillock, and then there are sodium-potassium pumps along the entire axon. As a result, propagation takes place. When an action potential occurs, then it triggers, the sodium enters the axon, diffuses from short distance just beneath the plasma membrane. So it diffuses, it comes in from the nearby, this is the plasma membrane. So all the sodium ions nearby will diffuse into the axon through the channels. The resulting depolarization excites the voltage gated channels. The next voltage gated channel, just immediate to it, gets excited. Sodium and potassium channels open and close as going forward along the way. So sodium moves in, potassium moves out, and this entire action potential and triggering of the impulse takes place one after the other as it goes along. Um, So one thing to remember here is action potential itself does not travel along the axon. Rather, it stimulates the production of a new action potential. So one action potential can initiate the next action potential, and which goes ahead and propagates from one end to the other. Um, so there is a little bit of a distinction between action potential from nerve signal. So when I say there is a propagation of impulse, I mean there is nerve signal that is moving from one to the other. Action potential is a phenomenon that is taking place at one point. And nerve signal is basically movement of these action potential, which is causing the next action potential and so on from one terminal to the other. So the nerve signal is a traveling wave of excitation produced by self-propagating action potential. So basically, the action potential is self-propagating and trying to move forward. So think of falling dominoes. Okay, um, so when the dominoes are one after the other, no one domino travels to the end of the line, right? But one domino, each domino pushes the other, and then pushes the next, and then pushes the next, and so on. The dominoes effect take place. So it's very similar to that. So it's not like the action potential itself is traveling, but it is causing the next action potential to be initiated and to be completed, and then the next. And then the next, and so on, as a result, which causes the nerve impulse or propagation of impulse to take place. So this actually helps to understand how it takes place in the myelinated neurons. This is when it is unmyelinated neurons, where there is no myelination across the conduction. One other thing to remember here is that when the this propagation of impulse in unmyelinated neurons is slow, because it has to, the entire process has to take place one step to the next, and so on. So next comes for myelinated neurons. So before we go to that, some of the major factors that influences spread of the action potential along the membrane, it depends upon the action structure, axon structure, how much of, what is the width of the axon, that's very important, the diameter of axon. Um, uh, it depends upon the path of the positive charge inside the axon, across the axonal membrane. And of course, as I said, it depends upon the diameter and number of voltage gated channels that is present in the axon. So these are some of the major factors influencing the action potential conduction. So in case of you know myelinated neurons, and we have done this before, that this is an axon, as you can see, and this is the myelination. These are basically lipid like structure that are made by oligodendroids in CNS and Schwann cells by PNS. If you remember, we did how they differ in the way they myelinate the neurons. In this, the propagation happens little different. Remember, the myelination of the neurons does is not continuous. There are gaps in between, and there's a specific reason why there are gaps. Okay, these gaps where there is no myelinations are known as the nodes of Ranvier. This is the place where the voltage-gated sodium channels are concentrated at the node. The sodium-gated channels are present in here so that sodium can move into the axon at these places. So something to remember in here, which is very important, is the voltage-gated ion channels are scarce in myelin-covered internodes. Fewer than 25 micrometer square in these regions, so there are very few in myelin covered regions. So these region in here, the number of voltage gated sodium channels are less, but near the nodes of Ranvier, they are about 2,000 to 12,000 per micrometer square at the nodes of Ranvier. So these are huge number of voltage gated sodium channels. Mm, so there would be little point in having ion channels in the internodes. Why? The reason why they are so less in number is that because myelin insulates the fiber. So you can see these myelin shades are insulating the axon, and sodium cannot pass through the extracellular fluid and come to the axon anyway. So having more of these sodium channels in this region does not make much sense. When sodium enters the axon at the nodes of Ranvier, when it is entering at the nodes of Ranvier, it diffuses for a short distance along the inner face of the membrane. I mean, this inner face of the membrane, it moves. um, each sodium ion has an electrical field around it. When one sodium moves from one to the other and so on, it actually causes the sodium ion has an electrical field. And when one sodium moves to the another, the field actually propels that. Basically, it is actually activating the next sodium in the nearby. And when it moves from one node to the other, if you look at this node of Ranvier and this node of Ranvier, one sodium ions, when it comes, it will actually move past this region and try to influence the sodium ions in the next nodes of Ranvier. Each sodium ion has an electric field, remember. Each electrolyte sodium ions, it's a charge, so it has an electrical field around here, which causes it actually transfers the field to the next one and propels it. And so since it repels each other, the sodium ions and sodium will repel each other and will push this sodium ion to the next to node of Ranvier. Okay, so basically it's pushing or it's jumping. We sometimes say it jumps from one to the other. So this movement of sodium from one to the other, it looks like it is jumping the propulsion, or the stimulus jumps from one to the next, to the third, and the fourth, and so on. Since this propagation is um, happens so in rapid stages. We'll do this. Just give me some. So when it moves from one node to the next to the third, this jumping of impulse. It looks like the impulse is jumping from one node to the next, and that movement is known as saltatory conduction. Okay, so this movement is so fast that it occurs. The movement of um, the action potential that occurs only at the node. This mode of conduction is so fast that it can take place. The movement can take place in milliseconds. Um, so, so the saltatory conduction is based on a process that is fast in the internodes, but remember, it is important that when it moves from the one nodes of Ranvier to the next node of Ranvier should also have voltage-gated channels. One is multiple sclerosis, the other is Guillain-Barré syndrome, which are both when the myelin sheath is disrupted, it can cause several diseases associated with it. So we know in unmyelinated fibers, there is step-by-step depolarization of each portion at the length of the axonema. And for saltatory conduction, it happens at the nodes of Ranvier depolarization, where there is high density of voltage-gated ion channels. And it carries ions long distance, and it doesn't decrease with distance. That is, I mean that, individual influence can be that it can be detrimental from one node to the other, but since there are several internodes, it will jump the propagation from one node to the other for it to have its influence. So this actually shows you the difference between myelin sheath and unmyelinated propagation. So as you can see, this is a neuron which is unmyelinated, and this is a neuron which is myelinated. So when in the axon hillock, there is huge sodium-potassium channel present in here, which triggers the action potential, and then there are sodium channels that are present at each unit, and then this stimulus causes the stimulus of the next, and so on. Current flow due to opening of sodium channels take place, and this is how the movement is taking place from one to the influencing the next neighboring axolema, and so on. And so it's much slower. In case of myelin sheath, and the internodes are filled with sodium-gated channels. And so the neuron here influences basically the entire nodal place where it is myelinated. There are very few sodium-gated channels here, but the sodium-gated channels at the internode is a lot. So there's tons that are, in the internode. And so sodium is hopping from one place to the next, and so on. So the axon diameter and propagation of speed, pulse of propagation depends upon the axon diameter and myelination. Type A are the largest myelinated axon with a diameter which range from 5 to 20 microns. Or micrometers, you can say. These travel at a speed of 130 meters per second. It varies from book to book. It can be 130 to 150 or 120 to 130 meters per second. B fibers are 2 to 3 micron meters and travels at 15 meters per second. It is usually found in the visceral sensory and autonomic preganglionic neurons. C fibers are 0.5 to 1.5 microns and travels with a speed of 2 miles per second. These are unmyelinated sensory and autonomic motor. The advantage of myelin becomes clear when we compare A with the C. Okay, if we compare A with the C, note that the diameter increases tenfold, but the propagation speed increases about 120 times. So even if the diameter increases, but in here, the diameter is increasing tenfold, but the propagation is actually increasing almost 100-fold, so which is a huge advantage. So so that means the diameter has a huge influence on the propagation of impulse. So we had started with this slide, and I want to wrap up the entire lecture with that. So just to let you know that, if you remember, we had started that this person steps on the nail, and there's a reflex action that take place, and eventually we see tears coming up from his eyes. So whenever there is an action that takes place, there's a reflex action that occurs, and sometimes this reflex action can happen even that response can happen even before the brain is able to perceive it, but eventually it does. The initial stimulus happens, which goes into graded potential, threshold action potential generation. There's propagation of action potential, synaptic transmission in the spinal cord, reflex activation takes place, motor action potentials happen, and ascending pain pathway causes tears to come out. The pain receptors are involved. As a result, this entire process happens in order for this milliseconds or few seconds of action to